Hey guys, so today I did not get to near the stuff that I thought we would get to. So it appears that we're still having a little problems identifying parts of the scientific method, okay? And so what I mean by that is there's still a lot of trouble with identifying what the independent variable is, the dependent variable, what are constants, what's a hypothesis, what's this experiment even about. It really seems like we're in the classroom, there's just it seems like a lot of us don't know exactly what the experiment necessarily even is. We just read it and that's about it. We read it and we don't understand what's going on. So all we did in class today was really we dove into two of these experimental scenarios, and then at the end of class, I gave the kids time to work on the third scenario with the compost and turn that into teams. Or that's what you guys are going to do. They're turning it in person. But anyways, let's go over this experimental scenario number 1, okay? And so first we're going to see what exactly the problem / observation is, which I'm not too terribly worried about because sometimes it's really hard for you to try to figure out what the observation here is. But it's easier to come up with the question, and I'm fine with that. So let's go ahead and read into this. A student investigated whether ants dig more tunnels in the light or in the dark. She thought that ants used filtered lights that penetrated the upper layers of the earth and would dig more tunnels during the daytime. 10 ant colonies were set up in commercial ant farms with the same number and types of ants per ant farm. The same amount of food was given to each colony, and the colonies were the same temperature. 5 of the colonies were exposed to normal room light, and 5 were covered with black construction papers, so they did not receive light. Every other day for 3 weeks, the length of the tunnels was measured in millimeters using a string and ruler. Get rid of that. Averages for the light and dark groups for each were measured and then computed. The averages are listed in the following chart. Okay, so what's the observation? What's the question here? Or what's the problem? Ants dig tunnels. That's about as good as observation as I can make. That's why I really don't care for observations sometimes, but that's about it. But a better thing is the question. So what's this girl wanting to do with this? Well, she wants to know, a good question would be, do ants dig more during the daytime or during the nighttime? Okay, and you can pause this video and write that down. Do the ants dig more, or do they dig longer tunnels during the day or during the night? Okay, and then what your hypothesis does, okay, is our hypothesis, and maybe you want to write this down too on your graphic organizer or something like that. Maybe I should look at this one. Okay, your graphic organizer, right on the side or something. Hypothesis attempts to answer your guys' question. So let's attempt to answer this question. And something that I put, and I think you guys what you should do before you maybe watch this video is try to do it on your own really fast. And just get on here, and I would go to here, and you guys can type some stuff up. Okay, and I guess I can do that right now. So what's our question here? Do ants dig more tunnels in the light or in the dark? Question mark. Okay. Our hypothesis, we're going to attempt to answer it with an if and then because statement. So if there is more light, then the ants will dig more because they will be able to see. And that makes sense, right? The ants will be able to see because there's light outside, so let's go ahead and go with that. The procedures. I'm not going to waste time going ahead and rewriting these. For number 3, I will make you write out the procedures, and that's not you copying and pasting the procedures from up top. I want you to paraphrase these procedures here, okay? And it's not too hard. It's just paraphrasing what they have going on here. What happened was she took 10 ant colonies, and she divided up 5 over here, and she put cardboard paper on those guys to block out the light, and took the other 5 and put them in the light constantly. And then she checked the distance of the tunnels every other day for 21 days. Right? She used a string and a ruler, and then she took an average of those 5 that she had underneath in the dark, and she made the averages, and she took all the 5 that were in the light, and she made the averages for those guys, and then assembled it. But we don't talk about her putting it in the data table, because that's really not part of the experiment. Like, this whole scientific method is collecting data, so you don't have to say, I then organized into a data table, because you're actually doing that in the next section. That's not the procedures, okay? So, moving on from there, the independent variable. Okay, so what would the independent variable here? The independent variable is also known as the manipulated variable. Okay, this is the one that the scientist manipulates. He changes or she changes themselves. So in this, what is it? Well, it is light or dark, right? Are you putting the ants in the light? Or are you putting them in the dark? And that's what the scientist chose to do. Now, the dependent variable, this guy's also known as the responding variable. Okay, so this guy responds to the independent variable. Let me get it spelled right here. So the dependent variable responds up top here. So what's the good responding variable? I would say length of tunnel. Right? So we don't know how long the tunnels will be, because the light or the dark could affect that. So that's why we called it the dependent or the responding variable. The controls / constants. So anytime that you see the same, or something like that, that's a good chance that that's a constant. So let's look at a couple here. It says the same amount of food was given. So we'll put amount of food. What else? Let's see. Same amount of food. The colonies were at the same temperature. The same ants per colony. Not that kind of ant. Ants. And what else? The same number of ants per colony. Right? These need to be constant, because if we give one group more or less food, that could definitely affect their work. Right? They need food to eat. Sometimes they may overeat, or if you starve them or something like that. Same temperature. If they're too cold or too hot, that could definitely throw off the results. Same ants. If you get a different ant colony that definitely digs more than the other, or the same number of ants per colony. Obviously, if I get 50 ants in this colony and 100 ants in the other colony, the 100 ant colony is probably going to dig more. Okay, and you guys can go ahead and just make sure you're reading along with me so you can see this, and then maybe writing this with pen and pencil, or switch back and forth, and maybe doing some pausing. The control group, there's not a control group here. I would say the control group in this could have been, but they don't have one here, is something that we don't change at all. And so that would be, we use an ant colony that we put outside, so it experiences daytime and nighttime just like a regular ant colony would. So we can then see with the ants that are naturally going through daytime to nighttime, maybe they dig more. Okay, maybe they need that dark as well as night or something like that, but they don't have that. Experimental groups. What are some experimental groups? Well, that would be the light. We're testing the light and dark ants. Okay, light, dark ants. So now we have all of these guys filled out. You should have your procedures filled out. So now we're going to go ahead and go to our data and results, which I have right here. So there's the effects of germination on radiation. That's the next one. But here we go. So what I have here is we need a title, right? So a good title for this one is the effect of light on digging. So this is a line graph. Okay, so a line graph is, we use a line graph to study the effects of things and compare them over time. Okay, so it may be day, it may be a day, it may be an hour, maybe minutes, seconds, whatnot, time, whatever. Okay, so an independent variable you could say is day, right? Because the scientist chose that he was going to measure every other day and chose that he would end it after 21 days. Another independent variable was the dark and the light, because the scientist chose, right, to put 5 groups in the light and put the other 5 groups in the dark. Our distance, Doug, this is the dependent variable. This is the distance where, or the variable that definitely responded to whether it was dark or light. Okay, and we can see this. And so obviously if I see my dark here, which is in red, I have my key up here. That's my key. I didn't put key or anything. Maybe I should have. I should have drawn a box and then wrote Kia here. Maybe I'll do that next time. Anywho, but if you guys are looking at your sheet again, and I'm looking at my data table just to kind of go through this first one, if I'm looking at my dark ants, so I go over to day 1 right here, and then I go up to 7. So I know I have 5, and I go into skip a line, and then I go to 15. Skip a line, I go to 25. So really I'm going into 5 millimeter increments, but I just put it every other 10 because it makes my side of my graph here much more neat, so I'm not cramming in a whole bunch of numbers here. Okay, and so something else I did mess up on that I did not realize, so my highest number is actually going to be 136, and my last number is 135, so I should have gone one more, and I went up to 145, so that's my fault. But if you're doing this, go to 145. Okay, so anyways, my first number is on day 1 in the dark. It's 7. So I go 1 over, up 7. Okay, then I go to day 3. Day 3 says 15 millimeters. So then I go day 3, 15 millimeters, right there where the 15 millimeter mark is. They intersect, right? Then I go over to day 5. Day 5 is 25 millimeters. So I go up here. There's 25. Day 7. I go up here to 32. Day 9, I go over here, and then I go up to 47. From there, I go to day 11, and I go up to 62. Okay, which is right below 65 there. I go over to day 13. Then I go up to 93. Big jump on that one. So I'm at 93 here. Then I go to day 15, go up to 110. From there, I go over to day 17, jump up to 115, go over to day 19, jump up to 120. Then I go over to day 21, and I finally go up to 136. Okay, and that's the same exact process that you're going to do with the light as well. And I don't have to read that off to you, okay? And then you connect the dots with your pen or your pencil, whatever you're going to do, okay? And I would like you to go ahead and take a picture of this, see if you can scan it. If you want, I know a couple of you know how to do this on Word. You can actually type this in on Word and come up with a line graph. Okay, that's actually right. If I want to go to Insert, Chart, and I go to Line Graph. Okay, and here's your line graphs. If you guys want to do that, you're more than welcome. You have to type in your data, and once you type in your data and everything, it actually works pretty well. And then you can actually you can drop that off in the Teams box. And remember, this is due. When is this due? September 17th, so on Thursday. Okay. For some reason, I had the 9th in my head, but that would be very wrong. Okay, so that's where you go here. So anyways, here in our conclusion, we'll go ahead and talk about my hypothesis. Let's see here. Go back to home here. Write in red so you guys can see what I'm writing in. My hypothesis was rejected. The ants in the light only Doug 103 millimeters in 21 days, where the ants in the light dug 136 millimeters on average, right? This is averages between 5 other colonies on average in 21 days. Okay, now it would have been kind of nice to see their raw data to see if maybe there was a colony in the light that did dig more than one colony in the dark. That'd be nice to see, but we're in the 7th grade, so it's okay. Okay, so we addressed our hypothesis. It was rejected, and here's our proof. Okay, I didn't say I have evidence. I didn't say I have proof. I actually put the evidence and proof in here, so I don't have to go ahead and literally say I have evidence and proof. I just put the proof here. Okay, and then I'll go ahead and also talk about any observations that I noticed. So like I was talking to the other kids, there is a time where they go up a little more. So you could say maybe from like day 11 to day 15 for the ants in the in the dark. Okay, they started digging a little more. Just little observations like that can lead to you going off on doing another experiment. So the ants in the dark began to dig more than the other days between day 11 and day 15, or it's like day 7 and day 15, you could say. Okay, and then we'll just kind of leave it there. But maybe if you're doing your own experiment, you would go and try to research, like, do these guys run in cycles? Do they work for 5 days and maybe take a break for a couple days or something like that? And this is where you infer, why do you think that is? Okay, and really we don't have anything to back that up or really know why. All right, so let's go ahead and head back to, so that's it for that one. So if pretty much what you should do is just look through this. Did you get the same answers that I did? If you did, then you probably don't need to listen to this whole video. Okay, you should probably keep listening if I'm going to go ahead and do number 2. So you can just skip forward to where I get to number 2, which is the end of the video, and check my answers. And if your answers are the same, if they're not, then you should rewind and try to figure out where you went wrong with 1 or 2 or how many ever you got wrong. Okay, but I'm going to go ahead and go on with experimental scenario number 2 here. Okay, so when a student investigated the effect of radiation on germination of bean seeds, he thought that the exposure would limit the seeds' ability to germinate, which means to grow right from the start. Not germination is not when it actually keeps growing into a stalk or anything, but just right from the seed. Much like the ultraviolet light causes skin cancer. 300 seeds were soaked in distilled out water for 1 hour. They were then divided into 3 groups. One group was placed in the microwave oven on high for 3 seconds. The other group was microwaved on high for 6 seconds. The last group was not microwaved. The seeds were then planted in 3 seed flats, which are just raised plant beds. And given the same amount of water, the seeds were planted in 3 separate flats. Given the same amount of water, the flats were placed in the location with a constant temperature of approximately 27 degrees Celsius. So each day for 2 weeks, the number of seeds that germinated each group was recorded. All right. So we can see that the 3-second group, these guys were put in there for 3 seconds, 54 of them germinated, meaning they pretty much sprouted. And they did this over 2 weeks. And the 6 seconds, they put them in the microwave. That was 26 that actually sprouted or germinated. And then no radiation. So they just they distilled them in that water, or they put them in distilled water for an hour, and then they just planted them, and 88 of them sprouted. Okay. So let's go ahead. What's our question here? Let me see. How does radiation affect the growth of bean seeds? Okay. Well, what's our hypothesis? Our hypothesis, right? We're going to answer our question. So if we put the seeds in the microwave for a longer period of time, then they will then fewer bean seeds will germinate. And remember, germinate just means to sprout right from the seed. Because UV radiation can cause damage to our skin. Okay, so when you hear radiation, then you hear ultraviolet radiation. That gives us a sunburn, right? Can cause damage to our skin. So maybe this microwave radiation. So when you guys hear microwave, you're like, well, that's the thing in my house. Yes, it is. But there's also this radiation that comes from the sun called microwave radiation. So that's why your microwave is called a microwave. Again, you're going to put your procedures here, and you're just going to paraphrase the what they have in the paragraph. Do not copy and paste that. What's our manipulated variable? What's the one that the scientist manipulates? The independent variable. I would say the amount of radiation exposure. All right, so we have none, 3 seconds, and 6 seconds of exposure. So then what's going to respond? Number of seeds that germinate. Okay, what's there some constants? I believe the amount of water, same number of days to grow. So she picks them up, or whoever does this picks them up after 21 or after so many days. After, what was it, 2 weeks? Sorry about that. Same amount of water. That. What else? Probably the same microwave would be a good idea. Both times the microwave was placed on high. Let's see, the temperature was the same. All right, and I think that's it. They're all soaked in the same distilled water for an hour or two. Control group. So we do have a control group here now. This is the group that we're running the experiment against to see if anything was actually changed compared to the normal. So our control group here would actually be the non-microwave guys. Microwave seeds. Okay. Then our experimental groups down here would be the 3 seconds and 6 seconds groups, which are actually our experimental groups, right? So we would like to know if radiation even affects them. So we need to go ahead and check out if the microwave, if the seeds aren't put in a microwave and they grow the same as these experimental guys, then we don't even know if radiation affects it. Okay. And up here, we can go ahead and see that the no radiation, this is our control group here. The no radiation guys, much more of these guys were actually germinated. The 3 seconds, the guys who were put in the microwave for the least amount of time had 54, and then the guys who had the most exposure to radiation had 26. So let's go ahead and see what this looks like. Okay, so we're going to go ahead and scroll on down, and this is what your graph should go ahead and look like. So here's my dependent variable over here. So number of beans that germinated. I went by tens. Skipped a line 10, skipped a line 20. Okay, so here's my exposure to radiation. There were 88 of them, so I went up to just below 90, 88. Here's my 3-second guys, which was 54, so that's just below the 55 mark. And then I have my 6 seconds, which is just above the 25 because that's 26. All right, and here I didn't really need a key because I have everything perfectly labeled and I didn't need a key for this. I put radiation exposure down here, and that looks good. Now, if you had a key at the top and you put none and you didn't have none down here, let's say you got rid of none, 3 seconds and 6 seconds, and you put that up in your key, and then you gave me a different pattern or a different color for none, 3 seconds and 6 seconds, that would also be fine. But you just make sure you do have radiation exposure down here to make this graph correctly. Okay, and that will get you a good looking graph like that. All right, so what's something we can talk about here? Was our hypothesis was our hypothesis accepted or supported? I believe it was supported. What was it? If we put the seeds in the microwave for a longer time, then fewer seeds will germinate. So I believe that was true. We put them in there. The group that we put in there for the longest amount of time only had 26, and then the group that was in there for 3 seconds had 54, and then the group that didn't have any at all was at 88. So I would say that's some pretty good proof. And so I need to prove that proof here, and I need to talk about it. So my hypothesis was supported. The control group that was not exposed had the most with 88 seeds germinating. But then that was our control, right? The group that was in the microwave for the least amount of time, which was 3 seconds, had 54 seeds germinate, and the 6-second group had the least amount germinate at 26. So why was this? So this is where we can start inferring. We can start reading in between the lines. Well, we said something about the UV radiation, right, of the skin. And that was also said up here. We know that exposure to radiation would limit the seed's germination, much like ultraviolet radiation light causes skin cancer. Okay, so that's kind of where we got that from. So then instead of just using that general knowledge, if we were doing, like I said, a real experiment, like a project in class, you would go ahead and do a little bit of research on microwave radiation. Well, what do we know? Can you put a pot of coffee, or I don't know if you've ever warmed up coffee, hot chocolate, obviously maybe some soup in the microwave, and it's made it hotter, okay? So if we put something in the microwave and we heat it up and we give it this energy, we could talk about how that would then tear apart the cell and maybe make or you put popcorn, maybe talk about popcorn, right? In a kernel, there's a little bit amount of water. And then when you heat it up, that water can't be contained anymore because it has so much energy and it's getting so hot, and it pops the popcorn. All right. So it has a lot to do with that, but that's where we'd go and get research, and we'd look up some articles and some magazine articles and talk about that on here. And then we'd go and cite those guys, okay, and to back up your claims. All right, but that's how that should look. So what you should do for a grade. So I want to see all of this done. We did all of this in class, but what I'm going to grade is the compost one, okay? And I'll go ahead and tell you guys, this is also a bar graph. This is also going to be a bar graph. So let me know if you guys need any help at all while you're doing this. See ya.